Welcome to tonight's lab, which is on gas exchange in plants. This is Biology 103, Lab 9. My name is Professor Willis, and let's go ahead and get started so you can see what we are doing this week. Before we begin with the lab, I thought I would just review some of the basics on plants and plant anatomy. So you obviously probably know that plants perform photosynthesis. They take in carbon dioxide and they release oxygen during the process of photosynthesis. However, you probably might be asking yourself, well, how do they do this? Well, in their leaves, they have these little pores, and the pores are called stomata. So these pores will open and close. They will open so that you can take in CO2 if you're a plant, but when you do open, you do have the potential to not only obviously release your oxygen, but you can also release water. And that process is called transpiration. So it's one way that plants can actually lose water. So plants have to really regulate when they should open and when they should close these stomata. Do they really need CO2 in exchange for a loss of water? Something that plants have to deal with in nature. Here are some pictures of these stomata. So remember, stomata, they are pores. So they're holes in the leaves, and they are definitely where CO2 can come in from the atmosphere. And remember, carbon dioxide is needed for photosynthesis to run. And of course, this is also where water exits as well as oxygen. If the plant is experiencing high rates of transpiration, so that means they are losing a lot of water, that tends to correlate with them having lots of stomata. That means they have a lot of these holes on their leaf and they're open, and so water will leave these holes when there is less water outside the plant than there is inside the plant. Some things that scientists have researched are why do plants have different numbers of stomata, right? Is it because the plants are directly in the sunlight? Does it have to do with the color of the leaf? Does the plant store a lot of water or does it release a lot of water? Like what kinds of environments are they in? And do the leaves grow all year or do they drop them and release them in the winter? So there's a variety of things that can happen in the environment that control the number of stomata that occur in a plant. So your challenge for this week is you are going to try to figure out, will blade-like leaves contain more stomata than broadleaves? And what do I mean by that? So blade-like leaves are usually thinner and skinnier in shape. If you compare that to a broadleaf plant, they tend to have really big flat leaves. So you are going to be looking at 5 different species on this online lab, and you're going to be counting their stomata to see if you can come up with a pattern for the distribution of stomata. Those 5 plants that you're going to be working with are on the screen right there. So you have tropical sage, you have marsh hay, spiderwort, bluegrass, and tickseed. So all of those you'll be observing underneath a microscope to see how many stomata they have. The technique that was used to count the number of stomata on the plants is the one I'm going to describe here so you can understand where those pictures are coming from. So what you do is you take a leaf and you turn it upside down, okay, and then you paint that leaf with a medium to thick coat of clear nail polish and you let it dry. After that, you simply put a piece of tape on the clear nail polish and you pull the nail polish off of the leaf. Now, what that did is it created a print of the leaf, specifically the print of the stomata. Then all you have to do is put that piece of tape that contains the nail polish stuck on it on a microscope slide and put it underneath a microscope. So you're going to see this online lab already did that for you. All you have to do is turn on the microscope, place the slide underneath the microscope, and count how many stomata you see for each species. For this online lab, like I was just saying, you are simply going to take the microscope slides that have already been created for you on the online lab and you place them underneath the microscope. And then you simply just count the number of stomata that you see in the slide. And again, you can tell what the stomata look like because they look like little holes, okay? After you're done collecting the data, you're going to go ahead and create a claim based on the type of plants, broad versus blade, and their stomata number. Okay, and did you notice, was there a pattern in what you saw? And again, if you don't see a pattern, it is okay for your claim to be there was no difference in broad or blade-like leaves. I took a screenshot of the data table so you can see what you're going to be filling in. Also remember to take the average of the 3 fields. So just move the slide around and look at different places on the slide to get different fields. And then lastly, I will just suggest making sure you answer all your questions using complete sentences, type up your responses, and submit your lab to Blackboard by next week. All right, that concludes this week's lab. Good luck, and I will see you next Monday.